So, Dr. Muralidharan, shall we begin?
Yes. Can you still see my screen?
Dr. Muralidharan, can you hear us?
Yes. Can you hear me? Somebody has muted me. I don't know. Hello?
We can hear you.
Okay, thank you. Okay.
Dr. Muralidharan, can you hear us?
Yes, I can.
Okay, so if the sound is okay, so let's begin. Am I audible, Shonak? Can you hear me?
Yes, you are.
Okay, fine. So let's begin. So welcome all of you for today's 19th lecture of Progress and Prospects in Biology webinar series. And today we have Dr. Bhavana Muralidharan from Institute of Stem Cell Science and Regenerative Medicine from Bangalore for delivering today's talk. So before we begin, a few housekeeping things to be announced. All participants are requested to please keep your audio and video muted to preserve the bandwidth and ensure uninterrupted streaming of the webinar talk. And please feel free to ask your question by typing in the chat box, and all those questions will be moderated and answered towards the end of the session. So I would like to start off by introducing today's speaker to all of you. Dr. Bhavana Muralidharan did her bachelor's and master's in biotechnology with distinction from Guru Gobind Singh Indraprasth University, India. She did her PhD from National Center for Cell Sciences under guidance of Dr. Vasudevan Seshadri. She then joined Professor Shubhatule's lab in Tata Institute of Fundamental Research as a DBT Wellcome Trust Early Career Fellow for her postdoctoral research. She went on for her further postdoc research in Professor Adrian Isaac's lab in UK Dementia Research Initiative at University College London. She then came back and started her own lab, and she is currently an assistant investigator and DBT Wellcome Trust India Alliance Intermediate Career Fellow at the Institute for Stem Cell Science and Regenerative Medicine, which is also popularly known as INSTEM in Bangalore. She has received numerous prestigious grants from DBT Wellcome Trust, India Alliance, and EMBO and DST ACRB Government of India. She has received travel awards from Society for Neurosciences and International Brain Research Organization. She has also been invited to deliver talks at many international meetings. She has also been involved in supervisory role in COVID diagnostics in INSTEM Bangalore. She is also actively involved in science outreach activities, and she has been actively involved in National Center for Biological Sciences, the Human Body Inside Out initiative, and she has also been associated with Chai and Why seminar series, which is a very interesting seminar series, which is jointly organized by Tata Institute of Fundamental Research and Prithvi Theatre. So with that, we extend a very warm welcome to you, Dr. Bhavana Muralidharan, to deliver today's webinar talk on our platform. And we all look forward to get more insights about the larger idea of building the brain. So with that, over to you, Dr. Bhavana Muralidharan.
Thank you so much, Dushyant, for the introduction, and thank you so much for inviting me. I feel very happy to be presenting my work today here. I'm sure all of you must have seen this image of the brain. This brain in the human brain has about 86 billion neurons and about 1 trillion glia, which all interconnect with one another, and each of them are of different kinds. Because we seemingly interact with the world around us, we don't realize that all of these numbers, the trillions and trillions of cells, have all been made from scratch, and this entire process is very dynamic. So building of the neural network consists of several different steps. And it starts with a very basic process, that of the stem cells, which go on to generate all the different kinds of neurons and glia in the brain. So the stem cell divides and it generates more of its numbers so that it can sustain the entire development of the brain. And at point, just at the beginning of differentiation, or just at the beginning of neurogenesis in the brain, this stem cell undergoes an asymmetric division and gives rise to differentiated cells, namely neurons and glia. Further, these neurons and glia, the axons outgrow and they make connections, and that leads to the formation of a functional network. Needless to say, a mistake in any one of these steps leads to a range of neurodevelopmental disorder. And at the heart of all of this is actually the chromatin regulation, or the regulation which consists of transcription factors and chromatin regulators. So as you can see here in this slide, what we have is a stem cell.
Dr. Muralidharan, sorry to interrupt.
Yeah.
We cannot see your slides transitioning. So can you just share it once again?
Oh, okay. Sorry, you couldn't see anything. Maybe I'll start it again. No. How do I do it? I stop presenting and then
yeah, and then you share it again.
Okay. Is it fine now?
Can you do the transitions? Can you change this? Yeah, that's fine now. Yeah.
Oh, okay. Okay. Sorry for the glitch. So as I was saying, this is the... I'm sorry? Yeah,
it's perfectly all right. Yeah, sure. You continue, please.
Okay, thanks. So as I was saying, that this is the brain, and the human brain has about 86 billion neurons and a trillion glia of different kinds. And because we interact with the world in such a seamless way, we don't realize that, all these numbers have all formed from scratch, and this entire process of making the brain is quite dynamic. And the neural network all begins with a pool of cells called the stem cells or the mother cells, or we call them progenitor in the brain. And they divide rapidly at the very, during embryonic development, so that you make enough number of stem cells that can sustain the entire brain development. And at some point at the start of neurogenesis, they start differentiating. They undergo asymmetric division, whereby they make one stem cell and one differentiated progeny, and that differentiated progeny either becomes a neuron or the glia, depending on when it's being made. Then these neurons and glia, they put out axons, and then they connect with each other, and that leads to the formation of a functional network. All of these steps have to be executed perfectly well, because even a small mistake in any one of them can lead to a neurodevelopmental disorder. So as I was saying, at the heart of all of this is the stem cell or the progenitor cell. And the very first step in this process is fate specification. So as you can see, this very stem cell in the brain generates all these different kinds of neurons. As you can see here, the different kinds of neurons and the different kinds of glia. And if you closely look at what is happening inside the nucleus of the stem cell or the progenitor cell, you would see that a very dynamic process of regulation is happening inside it. The chromatin is being rearranged, and all of this happens with the crosstalk between transcription factors and epigenetic or chromatin factors, which regulate gene expression, which, if a protein is being made or not made, decides what kind of a neuron or a glia will be made, and that then leads to the formation of a functional brain. So at the heart of all of this is the chromatin regulation, which is the key to the diverse kind of cells which are being made in the brain. Two things are very, very important, and they stand out in this entire process, that of timing and location. So this progenitor when it makes a differentiated progeny, depending on the timing or the location, the kind of progeny which it makes, whether it's two different kinds of neurons or a neuron and a glia, all of this is different depending on when it is being made or where it is being made. So the two key aspects of this is when and where, namely the timing and the location. So today in my talk, I'm going to tell you about cell fate specification, which is one of the very first steps of making the brain. And the region I'm going to focus on, the region I work on, is the cerebral cortex, which is the seat of all higher order functions, namely language, learning, memory, and consciousness. And I'm going to tell you about the fate specification in two regions in this cerebral cortex, namely the neocortex and the hippocampus. The hippocampus is the seat, is the area where all your memory happens, and the neocortex is devoted to language, consciousness, thinking, and all of that. So I'm going to tell you about self-fate specification in the neocortex, where, as I was telling you, the timing of two different kind of neural subtypes are being regulated, whereas in the hippocampus, entirely different kind of cell is being regulated. The fate switch between a neuron and a glia is being regulated. So as I go on, I'll be telling you more about it. So let's start with the first part of my talk, that is self-fate in the neocortex. This is developing cortical primordium, and for people who not seen a brain, what is this? This is a section, it's called a coronal section. If you can see me in the video, you can see that it's something which is cut face-wise. Like if you cut sections like this is how you would see the developing brain to be. As you can see here, there are two key zones in the developing brain. This is at an embryonic day 12.5, which is 12.5 days post-fertilization of the zygote. And this is how the developing primordium looks like. And at this stage, mostly the primordium consists of proliferating. The area in yellow is the proliferating cells or the stem cells or the progenitors. And what you can see is actually only a very small sliver of cells which are post-mitotic or that which are neurons. So the question is how this primordium, which mostly consists of stem cells, go on to become this highly organized 6 layers of the neocortex. The neocortex consists of 6 layers of neurons, as you can see here, each showing a different color, and each layer of neurons has a different function, and I'll be coming to that in a bit. And the layers are 6, which are in the reverse order, that layer 6 is at the bottom, 5, 4, 3, 2, and 1. So how does this primordium, which is full of stem cells, become something like a very complex 6-layered structure? So as I was saying, it is all generated from the stem cells, and this process is highly regulated and timed. At embryonic day around 10.5, the ventricular zone is mostly full of stem cells. And the very first day, that is at 11.5, the stem cell divides and gives rise to the first layer called the layer 6. And here it is marked by a transcription factor, TBR1, and this is layer 6 here. The very next day in development, the very same stem cells switch and start producing a different kind of neuron, that of layer 5. And that happens at embryonic day 12.5. The stem cell, which was actually making layer 6 neurons, now switches to start making layer 5 neurons. These layer 5 neurons migrate past this layer 6 and go on and sit on top of it, and they form the layer 5. And here it is marked by the transcription factor FESF2. As you can see here, this is an adult neocortical brain. Layer 6 is here, layer 5 is here. The very next day in embryonic development, that is 13.5, this stem cell starts producing a layer 4 neuron. This migrates past both layer 6 and layer 5 and goes and sits on top of both layer 6 and 5. As you can see here in the green color, that is the layer 4 neurons. And in the adult cortex, here it is marked by ROR beta. So you can see the stepwise, gradation of this marking layer 6, here being layer 5, here being layer 4, and then finally by E14.5, 15.5, your two other layers, layer 2 and 3, the stem cell produces layer 2 and 3, which migrates past 6, 5, 4, and goes on, sits on top of it. And here it's marked by the transcription factor COX2. So one of the questions which we had was, what regulates this timing? And what if you, tinker or do something with this timing? What happens to the kind of neurons which are produced in the neocortex? To To understand this, we looked at the expression of a transcription factor, LHX2, which is a homeodomain transcription factor. Basically, it binds to a region in the DNA, which is AT-rich. So we looked at the expression of this transcription factor in the cortex to see whether this transcription factor could have any role in regulating such a timing of, regulating the timing between production of two neuronal subtypes, a layer 6, layer 5, or a 4 or a 3. And what we saw was this transcription factor at embryonic day 12.5, when it's still a lot of stem cells in the brain, is expressed in the entire, all the stem cells of the progenitor. So all of these are the progenitors, and all of them express LHX2. Very soon, 3 days later, at embryonic day 15.5, LHX2 still marks all the progenitors in the brain. As you can see, the brain has now developed from being a 12.5 to a 15.5 brain. And as you can see that the progenitor zone is starting to decrease as the neuronal zone, the zone which contains the neurons here marked by black asterisks, is increasing. But you would notice one thing, that LHX2 expression is still only in the progenitors, and it is missing from this postmitotic zone, which now consists of layer 6 and layer 5 neurons, and LHX2 is absent from this. It's excluded out from this zone. Finally, when we looked at a postnatal brain, the progenitor zone completely is gone by then. This is the 6-layered neocortex, which I showed you a couple of slides back. And you would still see that the deep layers, or the layer 6 and the layer 5, which I showed to you before, still exclude out LHX2, and LHX2 comes up postmitotically in the upper layers. So this made us, got us thinking that does LHX2 have any role to play in regulating the formation of layer 6 versus layer 5 neurons? And to study this, we used a mutant, mutant, where we very specifically removed LHX2 only in the cortex. It is driven by a Cree called EMX1 Cree, where EMX1 is again another transcription factor whose expression is driven slowly in the cortex. And we timed it in such a way that the removal of LHX2 happens right at embryonic day 11.5, which is the time when when the deep layer or the neurogenesis starts in the cerebral cortex. As you can see here, this is a control brain, and this is a postnatal cortex now. TBR1 marks layer 6. FESF2 marks layer 5. You can see this is 6, this is 5. I have put out 2 more markers for layer 5, namely ER81 and ET2. So now what happens in this LHX2 mutant, where we've removed LHX2 from the cortex at embryonic day 11.5? And what happens is that in this mutant, you can see that layer 6 is reduced with a concomitant or a simultaneous expansion or increase of the layer 5 neurons, as you can see here from the expression of FESF2, ER81, and ET2. So perhaps LHX2 is involved in regulating the fate switch between a layer 6 to a layer 5 neuron. And the moment you remove LHX2, what you see postnatally is that the layer 6 is reduced at the expense layer 5 is expanded at the expense of layer 6 neurons. We quantitated these results just to be sure that indeed the numbers have changed. And you can see here, this is an immunostaining. This is TBR1 layer 6, and this is layer 5 CTIP2. And you can see that, TBR1 is decreased, whereas layer 6 CTIP2 is increased. And these are the quantifications. You can see CTIP2 is increased, whereas with a concomitant decrease in TBR1. So what could be happening? Like, we have removed LHX2 embryonically at embryonic day 11.5, and we look at the postnatal cortex, we see that, one layer of neurons is increased at the expense of the other. So what could be happening? What could perhaps be the molecular mechanism? That was our immediate next question. And before I go on to tell you what could be the molecular mechanism, or what perhaps is happening inside the progenitors or the nucleus of the progenitors, I would like to tell you a bit more about what are the different neural subtypes and what do these subtypes do in the neocortical layers. So the layer 6, which is marked by TBR1, these are corticothalamic neurons. So when I say corticothalamic neurons, there is a region, all these neurons are supposed to project from one region of the brain to the other. So cortex is a higher center in the brain. Thalamus is a relay center in the brain. So these neurons, which are layer 6 neurons, they project from the cortex, from layer 6, and they project to the thalamus. Now, thalamus is a relay center. A relay center is basically what it does is it integrates all the, so whatever sensory modalities, input come from the sensory modalities like your touch or your vision or your auditory or anything, it all comes to the thalamus. And from the thalamus, it goes to the higher center in the brain, that is the cortex. Similarly, from the cortex, the layer 6 gives out information or gives output to the thalamus. And so therefore, the thalamus is a relay center. Now, the very next layer, layer 5, actually projects to a very different region. Now, this layer 5 has got two kinds of neurons. The first neurons, which are marked by FEZF2, CTIP2, are the subcerebral neurons. And one class of these neurons are your neurons which project to the spinal cord. These are called the corticospinal motor neurons. So what happens is from the layer 5, so all your movement happens because of these neurons. So the cortex then talks to the spinal cord via these corticospinal motor neurons, which come from layer 5 of the cortex to the spinal cord or other subcerebral regions in the brain. Now, this layer also has a different class of neurons called SATB2, which are the callosal neurons, which basically constitute or make this sort of projection neurons, which help you talk one part of the brain to the other, one hemisphere of the brain to the other hemisphere. So these are called the callosal projection neurons, which connect one half of the right half to the left half. So these are the different neural subtypes, and these are the different projections which they do. A bit is known about this switch, which as it happens from layer 6 to layer 5. So as I was telling you, say at embryonic day 11.5, when the progenitors are actually making layer 6 neurons, what actually happens is two transcription factors, TBRN and SOX5, they're very, very important for the making of layer 6 neurons. And they do so by actually inhibiting FEZF2. Now, FEZF2 is a very important transcription factor, which is for making of layer 5 neurons. So thereby, these two transcription factors repress FEZF2 to ensure that at 11.5, you only have layer 6 neurons being born and not layer 5. Just as, the timing proceeds from 11.5 to 12.5, two other SOXs, SOX4 and SOX11, literally knock off SOX5 from the enhancer region of FEZF2 and activate FEZF2, which in turn goes on to inhibit TBR1 and further activates CTIP2. And thereby now the neuron, which was earlier, which was making layer 6 neurons, has now started to make layer 5 neurons. And that's how you get the subcerebral projection neuron identity. Separately, some neurons of the layer 5, they express SATB2. SATB2 represses CTIP2, and that's how you get the two different populations in layer 5, namely the subcerebral projection neurons and the callosal neurons. So in this, we thought that what perhaps LHX2 could be doing to regulate this switch between layer 6 and layer 5. So we performed ChIP-seq because it's a transcription factor and it binds to target genes. So we should do chromatin immunoprecipitation to understand on which target genes does it bind. And we found that LHX2 is bound to both FEZF2 and SOX11, two transcription factors which are very, very important for making layer 5 neurons. And here I'm showing you the binding of LHX2 on the FEZF2. This is the FEZF2 entire locus. You can see the transcription start site is here, and this is the transcription start site of SOX11. And you can very nicely see that LHX2 is bound to the putative enhancer regions of both FEZF2 and SOX11. We validated, we indeed did see LHX2 bound at these regions as opposed to a region where there is no LHX2 binding site, thereby confirming that indeed LHX2 is bound at these regions. Now what happens is, so then we ask the question that now when we remove LHX2 at 11.5, how does this developing brain and the expression of FEZF2 and SOX11 look at 12.5? And as you can see here, this is a controlled brain where this blue is showing you the expression of FEZF2. It is very less in the progenitors, and it's just starting to come up in the postmitotic region. But as you can see, the moment you remove LHX2, there is an increase in expression of FSF2. And this is sustained even one day later at 13.5. As you can see here, the expression of FSF2 is still sustained in the absence of LHX2. Same is true with SOX11. This is the expression of SOX11 in the control, and this is the LHX2 conditional mutant. As you can see, there is an increase in the SOX11 expression. So thereby, this gave us an idea, an inkling about the mechanism that what LHX2 is perhaps doing is it's keeping the genes which are important for making the subcellular projection neurons, namely FSF2 and SOX11, repressed. At the moment you remove LHX2, this repression is removed and there's an up-regulation of these both genes, which then cells which were supposed to form the layer 6 neurons have now started to make layer 5 neurons. And that's why postnatally you see an expansion of layer 5 neurons at the expense of layer 6 neurons. We further wanted to understand, because this LHX2 is a transcription factor, it could be interacting with chromatin modulators or chromatin regulators for doing this. So we performed mass spec to understand the interacting partners of LHX2. And what we found was LHX2 was associated with a repressor complex, the NUD, or the nuclear remodeling repressor complex. And we were able to show that LHX2 was indeed interacting with specific subunits of this remodeler complex, namely LSD1, RBBP4, and HTAC2. And this is an IP to show that when we do LHX2 IP, when we pull down LHX2, we see that HTAC2, LSD1, and RBBP4, all three of them being pulled down along with LHX2. And we did reverse IPs. We performed HTAC2 IP, and we see LHX2 coming with it. We performed LSD1 IP, we saw LHX2. Similarly, we performed RBBP4 IP, again, we got LHX2. So now we have two things here, that LHX2 is interacting with, a complex, which is a repressor complex, namely HTAC2, and components of these, namely HTAC2, LSD1, and RBBP4. Also, what we have is LHX2 repressing two target genes, FSF2 and SOX11. So the next immediate question we asked was, now do these specific subunits, namely HTAC2, LSD1, and RBBP4, are they also bound at the same region as where LHX2 is bound? And also perhaps at the transcription start sites, because that's how the entire, chromatin regulation happens, where, distal enhancers, they come closer to the transcription start site via a looping mechanism. So we did in chips of LSD1, RBBP4, and HTAC2 to see whether they are bound at the transcription start site or the LHX2 binding bound region on the FSF2 SOX11 locus. And what we saw was LSD1 is bound both at the FSF2 locus and the SOX11 locus, both at the transcription start site and the LHX2 binding region. HTAC2 was bound at the LHX2 bound region and was not seen at the transcription start site of both FSF2 and SOX11. Interestingly, RBBP4 was only bound at the SOX11 locus, but not at the FSF2 locus. So perhaps what is happening is that, we may not have a complete holo NUD complex which is bound to some of these locus, but perhaps there is a dynamic interaction happening between LHX2 and some of these components. And depending on how it has to fine-tune the gene expression, some of these are being recruited to these enhancer regions or the transcription start site regions. So finally, we wanted to know that, yes, LHX2 is repressing FSF2 SOX11. These subunits are also present at the same region. What happens to the marks at these regions? And we wanted to know what happens to two active marks, namely H3K4 trimethyl mark and the H3K9 acetylation mark. Are these now, in the absence of LHX2, are these now going up? And that's how perhaps the expression of both FSF2 and SOX11 is increased in the absence of LHX2. And indeed, we found that both at the FSF2 locus, SOX11 locus, we can see H3K4 trimethyl mark going up at the transcription start site and the LHX2 bound region. And similarly, so is the H3K9 acetylation mark going up at the LHX2 bound region and the transcription start site of FSF2 and SOX11 locus. So till now, I've been telling you how LHX2 is a necessary transcription factor to regulate the neuronal subtype switch between layer 6 and layer 5 neurons. The question we asked was, is this sufficient to suppress CTIP2? Which means that if we overexpress LHX2, can we now repress or suppress the CTIP2 positive cell-fate acquisition? So to do this, we performed something called in utero electroporation, where what we do is we access the embryos, embryonic brains at inside the pregnant female. We do perturbations like electroporation, where we electroporate the either EGFP or LHX2 inside this brain. Then we stitch up the mother and let her develop normally. And postnatally, we harvest the brains of this embryos which were manipulated during development, and we look at we harvest the brains and we look at what happened when we either overexpress LHX2 or we overexpress EGFP. And these are just some of the images, and I'll show you the results in the form of a bar graph. So at embryonic day 12.5, a majority, 55% of the neurons are CTIP2 positive, expected because this is an age when layer 5 neurons are being born. The moment we overexpress LHX2, what we see is that the numbers are reduced drastically in the present when we overexpress LHX2. They're clearly saying that LHX2 is sufficient to suppress CTIP2. We ask the question that now this neuron, which can no more become a CTIP2 positive neuron, what is the fate of this neuron? And interestingly, what we saw was the fate of these neurons had become SADB2 positive neurons. So there was this, not only was LHX2 depressing CTIP2, but because there's a cross regulation between SADB2 and CTIP2, the neurons were becoming SADB2 positive. Right. To summarize this part of my talk, so what we have shown here is this LHX2 is bound to its putative enhancer region, and because the chromatin loops, and thereby it's able to thus access the transcription start site of both FEZ2 and SOX11, and because these interacting partners interact with LHX2, they're able to access the transcription start site and the binding region, and HDAC2 removes acetyl marks from the histone, and therefore you have very less active marks on these loci, and therefore LHX2 keeps these repressed. So the moment you remove LHX2, these are no more able to access either the enhancer or the transcription start site, and what happens here is that the active marks increase because HDAC2 is not now able to remove these acetyl marks, and thereby leads to an increased expression of FEZ2 SOX11, which then leads to increase or the expansion or production of more number of layer 5 or the subcerebral cortical projection neurons. So what we have shown here is LHX2. So I showed you this slide a couple of, at the start of my introduction, that, this complex molecular network which regulates the layer 5 to layer 6 neuron identity. And what we have shown here is LHX2 is repressing both FEZ2 and SOX11, and thereby controlling the switch from layer 6 to layer 5 production. So till now I was telling you about the timing. Is there a spatial angle to this? And perhaps there is, and I'll show you very, a brief inkling that perhaps there is a spatial angle to this as well. So this is a mouse brain, and this is from the rostral. This is the nose end, and this is the back. That is from the rostral and the caudal end. And different regions in the brain do different things. For instance, the front is the motor cortex, followed by the somatosensory, then is the auditory, and then is the visual. So different regions are devoted to different, perceptions. So the motor cortex is the region where, all these has increase or, maximum expansion of the subcerebral projection neurons or the corticospinal motor neurons which come from the cortex and projects to the spinal cord. When we looked at the LHX2 expression, and we saw that the expression of LHX2, so again here on the left side is the nose, or where eventually your nose will be, and the right side is the back of the brain. So if you look at the LHX2 expression, you will see that it is very well complementary to where the motor cortex is actually formed, and you can see LHX2 expression very high in the caudal regions. So perhaps LHX2 is is part of a regulatory network whereby you can control the production of motor neuron or the corticospinal motor neurons, and you can control the numbers being produced across the different regions in the brain. So this perhaps gives an insight into maybe there is a spatial regulation, and perhaps LHX2 is a part of such a regulation to control the number of neurons which are being produced in one part of the cortex as opposed to the other, right? So that was the first part of my talk, where I told you all about cell fate in the neocortex. What I didn't tell you in the very first slides when I showed you this cortical primordium is that this primordium, which is a cortical primordium, is actually consists of two regions which are developing adjacent to each other. The region in blue is the neocortex, which forms the highly complex six-layered architecture. But the region in yellow goes on to form the hippocampus, which is relatively a simple structure, consists of a single layer of pyramidal neurons. It doesn't have this complex six-layered structure. And it is also, it's the archicortex, and neocortex is primitive. It's more primitive as opposed to the neocortex. So when we looked at the LHX2 expression, we saw that LHX2 has a gradient of expression. It is expressed more in the hippocampus and relatively less in the neocortex. And we had shown a few years back that LHX2 in the hippocampus was regulating the neuron glia cell fate switch. So what is that switch? Is that in the developing hippocampus, very early during development, when the levels of LHX2 are high, these progenitors are producing neurons. As the time progresses in gestation, when all the neurogenesis has almost nearly completed, there is a fate switch, and this time the fate switch is between the production of two different kinds of cells. The fate switch happens, and the progenitors whose levels of LHX2 are low begin to produce glia. We had shown that when you remove LHX2 from these early progenitors, you lead to premature production of glia. But if you overexpress LHX2 in these late progenitors, you prolong the neuron production. And this fate switch was happening only in the hippocampus, but not in the neocortex. And I've told you just in my entire first part of the talk that in the neocortex, LHX2 is actually involved in regulation neuronal subtypes. Whereas in the hippocampus, where there's just a single layer of neurons, LHX2 is regulating the production between a neuron and a glia. So we didn't know about the mechanism by which LHX2 could function. So to understand this, I again performed ChIP-seq from this E12 developing hippocampus. So we very carefully dissected out the hippocampal primordium from the neocortex. And again, I performed ChIP-seq to understand that what could be the downstream regulators of LHX2. And what we saw was LHX2's downstream targets were two transcription factors, PAX6 and neuroc2, which have been previously known or shown to regulate this neuron glia cell fate switch. And we also uncovered a novel player in this neuron glia cell fate switch, namely DMRT5. And in the next few slides, I'll be telling you how, we looked at what is the sort of regulatory network these four transcription factors, they do to bring about this neuron glia cell fate switch. So the first thing, we looked at was what happens to the expression of these PAX6, neuroc2, and DMRT5 in the absence of LHX2. So we again took the LHX2 mutant. This is a wild type. As you can see that now this is the developing hippocampus. You can see it here. And you can see the expression. This is the expression of LHX2. And here I'm showing you two markers for the hippocampus, namely ZBTB20 and PROX1. You can see the expression of PROX1 here and ZBTB20. The moment we have a mutant of LHX2, the hippocampus is reduced, the size is reduced. But indeed, you could still see the expression of ZBTB20 and PROX1. But what happens to the expression of PAX6, NeuROC2, and DMRT5 is that all these three are highly diminished in the absence of LHX2. So these are the expressions of PAX6, NeuROC2, and DMRT5. In white is their expression in the progenitors of the developing hippocampus, whereas their expression is highly reduced in the LHX2 mutant. Now, so I developed a ex vivo electroporation assay to now ask do some of these manipulations ex vivo, that is out of the embryo, and probe whether the progenitor forms a neuron or an astrocyte. So what we do here is we take out the intact brain in a dish and we inject the DNA into the ventricle or the hollow space inside the brain. And then we target electrodes in such a way that it can be, the hippocampus can be electroporated. So after we do this, we maintain it as an explant, and then we dissociate these explants and seed them as cultures to ask the question whether they're making neurons or glia. So I performed these techniques and I electroporated, removed LHX2 from these brains. So these are conditional accord brains. So the moment you overexpress CRE, you remove LHX2. And in the same, if we overexpress NeuROC2, PAX6, and DMRT5, we can then ask the question that what happens when you remove LHX2, you see a lot of glia, but now is PAX6, NeuROC2, and DMRT5 overexpression able to reduce the glia production? I'll show you the images in a while, but I'll just summarize the results in the form of graphs. So what happens is at embryonic day 15, you have actually you only have 20% of the cells producing glia. So if you plot a graph, you would, and you electroporate EGFP in these cells, you see about 20% of them are being glia. But the moment you overexpress LHX2, PAX6, NeuROC2, or DMRT5, they all, in to varying degrees, reduce the production of glia, thereby telling us that, like LHX2, PAX6, NeuROC2, and DMRT5 are indeed neurogenic, which means the moment you overexpress them, they're able to change the fate from becoming a glia to neuron. And we did the same thing in the absence of LHX2. So this is electroporation of GFP, where there are 20% production of glia. The moment you remove LHX2 using a CRE in these LHX2 lox mice, you see 70% of the cells becoming glia now. So there is this switch, and there is a premature gliogenesis which happens in the developing hippocampus. And now in this condition, if you overexpress PAX6, or you overexpress NeuROC2, or you overexpress DMRT5, they are sufficient to rescue the LHX2 loss of function. So thereby saying that, they are indeed potent neurogenic regulators of this neuron glia sulfate switch. So here I'm showing you images of my cultures. As you can see, this is EGFP, and I'll show you images from two stark conditions of LHX2 removal and that of NeuROC2 overexpression. So this is from an LHX2 removal condition, where you can see that almost all of these electroporated cells here, which are LHX2 and GFP positive, they're all GFAP positive, and none of them seem to be beta tubulin, which is a marker for neuron. GFAP is a marker for astrocytes. Similarly, when you overexpress NeuROC2, you see that all of these cells are beta-3 tubulin positive and not GFAP positive. Okay, so as I told you, PAX6 and NeuROC2 were previously known regulators, but DMRT5 was something novel. So then I went to the lab of collaborator Professor Eric Bellefroid, who had these DMRT5 mutants, to understand that what happens when we overexpress LHX2 in a DMRT5 mutant. Is it still able to rescue the neurogenesis, or how's the regulation between DMRT5 and LHX2? So this is now a DMRT5 mutant brain. As you can see, that LHX2 expression is reduced in the DMRT5 brain. And likewise, in the DMRT5 mutant, where removal of DMRT5 led to an increase in production of glia, the moment we overexpress LHX2, it is completely able to rescue the DMRT5 loss of function. We saw that there is a evolutionarily conserved pathway by which perhaps LHX2 is regulating DMRT5. Now, this is the DMRT5 locus, where this is the transcription start site here. And we see that LHX2 is bound to a region which is conserved across species between Xenopus, chick, and human, and this is the mouse axis. So LHX2 is actually bound to the DMRT5 locus at this region, and this is here that locus which is conserved between mouse, human, and chicken. As you can see, that this is a very classic LHX2 binding motif, the TAATTG motif, by which LHX2 binds to its target loci. So thereby, what we have shown here is that LHX2 it's like a complex network where LHX2 is activating PAC6, NeuroC2, and DMRT5 in regulating the sulfate switch. Now, LHX2 and DMRT5 cross-regulate each other. LHX2 is able to activate DMRT5, DMRT5 is able to activate LHX2. What I have not shown you, and this has been published before, is actually DMRT5 represses both PAC6 and NeuroC2. So what we have uncovered here is perhaps, a complex biregulatory network where both LHX2 and DMRT5 have opposing roles in maintaining the levels of PAC6 and NeuroC2. And in doing so, they're maintaining critical levels of PAC6 and NeuroC2 to, carry out this process of, neuron glia sulfate switch, or maintaining the balance or keeping the glia repressed at the time of neurogenesis in the developing hippocampus. So this is the sort of my second part of my talk. So in my entire talk, I have told you about one of the very first step in cortical development, that is the sulfate specification, and how chromatin regulation, that is a crosstalk between transcription factors and chromatin regulators, plays a very important role. And I've given you examples that how, timing is very important, because the timing can determine whether you're going to make two different kind of neurons. The timing can also determine whether you're going to make a neuron or a glia. And all of this is also different in different regions of the brain, namely the hippocampus and the neocortex. And at the heart of this progenitor is this chromatin regulation, which regulates the expression of target genes and thereby leads to formation of a functional brain. And I'll end my talk here and finish with acknowledgments. All the work which I've shown to you is from my postdoc, which I did in the lab of Professor Shubha Tole. And I would like to thank her for her mentorship. And I would also like to thank Professor Sanjeev Garande and Professor Eric Bolefroid. Sanjeev at ICER Pune and Eric is at in University of Belgium. I thank them both. They're the collaborators on these, both the projects which I showed to you. And this is my current lab. Yes, we all participated at the COVID Diagnostic Center, which we have at INSTEM. So we are all proudly sporting the COVID Rockstar t-shirts, which they gave us. And my current lab is funded by the India Alliance, core funds from INSTEM and DST Serve. And with this, I'll end my talk and I'll be happy to take questions. Thank you so much.
Yeah, Dr. Muralidharan, thank you so much for the wonderful and engaging talk. And we really got some new insights into how actually chromatin regulates the brain development. And we have some questions in the GMIT section, so we can take them if you're okay with it. Shall we proceed with the questions, Dr. Muralidharan?
Yes, please.
Okay,
yes,
sure.
I was just, can you still see my screen?
Yes, we can see your screen now.
Okay, yeah, that's fine.
So the first question is from Kaustav Mukherjee. He wants to know, does spatiotemporal overexpression of LHX2 affect the cell fate determination process? If yes, then how?
Overexpression of LHX2? Yes, indeed. Okay, I can first answer temporally, right? So temporally, we overexpressed LHX2 in the neocortex. The in utero electroporation data which I showed to you. Can I go back to my slides?
Sure, you'll have to share it again, I guess. Yeah.
Is it shared now?
Yeah, it's shared now.
Okay. Sorry, I'll just quickly run back. So spatially here, we overexpressed LHX2 in the neocortex. And the timing was at a time when actually you have a lot of production of deep layers or production of layer 5. So the moment we expressed LHX2, we saw a decrease in C-tip2 positive. And the mechanism, I think we believe what LHX2 is doing is, it is interacting with the node remodeler complex, and it is recruiting some of the subunits, it's perhaps recruiting some of these subunits, key being the HDAC2, onto the target genes FESF2 and SOX11, and thereby repressing the FESF2 and SOX11 loops. And that's why we are not able to get layer 5 neurons here. So this is a very specific targeted expression of LHX2 in the neocortex. But now if we do the LHX2 overexpression in the hippocampus, we see that, and I think that's data which I've not shown, and it's published, it was published a few years back, but I can summarize it here. Yeah. So now if you overexpress LHX2 in, again, spatially you overexpress it in the hippocampus at an age when you actually get glia production, we know that it leads to the prolonged production of neurons. And this, I believe, it's because LHX2 is now activating transcription factors PAC6 and NeuroC2, something which my work has shown, that which are very key potent neurogenic molecules. So the moment you overexpress LHX2, it goes on to activate PAC6 NeuroC2, and that leads to the production of glia. So yes, spatiotemporally it is doing different things, and we are able to crack the mechanism. In the neocortex, we still don't know how LHX2, mechanistically how it's interacting with the chromatin in the hippocampus. We still don't know that. Does that answer?
Yeah, I hope that answers the question. And the next question is from Abhishek Bardhan. Have you considered the chances of involvement of non-coding RNAs like microRNA or long non-coding RNA in these well-regulated processes?
That's a fantastic question. I'm sure there is some non-coding RNA. We've not looked at it. We've not looked at it. I think we need to look for non-coding RNAs, because a lot of non-coding RNAs are expressed in the developing brain. So I'm sure there is some crosstalk happening. We've not seen.
And the next question is from Srikant Burgula. Madam, please can you explain how does chromatin structure affect transcription? How does the chromatin structure affect transcription?
So, okay. So these remodelers are, so the basic chromatin unit is a nucleosome, right? Where, the octamers of histones are wound around the DNA, and if it's equally spaced, the expression is regulated depending. So if you need to activate a gene, you need to sort of remove some of these nucleosomes, remove the octamers and open the DNA, and that leads to transcription. So some of these remodelers in the node have, subunits which actually translocate. They have ATPase activity, and that's how they translocate the DNA and open the chromatin, or shut the chromatin otherwise. So that is at a very local level that, some of these, how these remodelers, for instance, acetylation of histones then opens the chromatin, and the deacetylases remove the acetyl marks, and that's how it shuts the chromatin and represses the target gene expression. Now, how this happens long range is that, though we have not shown, but there are several techniques which show, namely the high C or the high chromatin conformation capture techniques, which show that, the entire thing plays out as loops. So they're chromatin loops, whereby distal enhancers, they come in close proximity to the DSS, and many different enhancers could be involved in such regulation. We think that one such mechanism is probably at play with LHX2, though we've not yet looked at any high C technique to say that that's what is happening.
And the next question is from Gaurav Bhardwaj. And he wants to know on 12.5 day, what factor suppresses the activity of LHX2 in normal condition?
Okay, that's again a very nice question. We don't know at 12.5, but we do know that there is another transcription factor called FOXG1, which regulates the expression of LHX. And that is slightly earlier in development. And we also know there is another, I think it's 6.3, another transcription factor. I think it's 6.3, which also regulates LHX2 expression. Some of these are very dynamic, right? So at E12.5, I cannot say what transcription factor is regulating the expression. We still don't know. I think we'll be very interested to know what regulates the expression of LHX2 at 12.5. Though there is a possibility, I mean, though we know that FOXG1 regulates LHX2, so does 6.3, and there are other transcription factors at other time points in development.
And the next question is from Prabhat Kumar Swain. He wants to know what is the duration of time for glia in neuron production as per your study.
Okay. So what happens is all over the mammalian nervous system, or in the case of the mouse development, the embryonic days from 11.5 to late 16, 17 is a neurogenic period. And by the time neurogenesis tapers off, and just right before birth, that is E17, the astroglia production increases, and it goes on till postnatal. So even after birth, the glia production happens, because there are different kinds of glia. It starts with production of astroglia, then oligodendrocytes, and so on. So the production starts at the time just before birth of glia, and then it continues postnatally.
Well, I hope that answers the question. And the next question is from Atanu Banerjee. What kind of consequences could happen if discrepancy happens in timing of neurogenesis and gliogenesis?
Okay, so in neurogenesis, fate switches are known to cause, for instance, I didn't talk about another class of neurons called the interneurons, right? So if there is an overproduction of interneurons, it has been implicated in autism disorders. So this fate switch is very, very important. The maintaining the critical numbers of neurons is very, very important. Whether there's any disorder which leads to, because of the fate switch of neuron glia, I'm not sure, because glia anyways are so huge in number that I'm not aware if there's any disorder which is caused by the neuron glia cell fate switch. But you need the right numbers of neurons, subtypes, otherwise in fact, even TBR1 has been, which is also important, as I was saying, was important in the layer 6 neuron specification. A TBR1 mutation causes neurodevelopmental disorder. So it is very important to keep all these, things very important. Like, for instance, the very first step in this entire process is progenitor proliferation, right? So if you have a mistake in that, that leads to the formation of microcephaly, which is reduced brain, a small brain. So you need these processes to be very stringently or robustly maintained. Otherwise, you'll have problems like neurodevelopmental disorders or things like that.
And Sanchari Gore wants to know, can adult brain stem cell be specified to different parts of brain for therapeutic approach using such studies? So
that's a very good question. So adult stem cells, in fact, people are, so adult stem cells do produce neurons, but there are very specific niches in the brain. So one such niche is the dentate gyrus niche, where you have stem cells which produce the hippocampal neurons. Whether you can actually reprogram some of these neurons and astrocytes also to produce different neural subtypes. And that's like a very upcoming field, because what people are trying to do is, if you can reprogram some of these neurons, so you can take out some of these neurons from, and locally reprogram them. So if you have some lesion in some part of the brain, if you can make the neuron already in that part to reprogram and make some other kind of neuron, that could be a very useful, has a very useful therapeutic potential. So yes, reprogramming is quite possible. Yes. Sorry, you're on mute.
Sorry for that.
Yeah.
So the next question is from Anushka Banerjee. Would brain organoid models provide any advantages in such studies?
Definitely. So I think, I mean, if I can modify the question, is that, I mean, organoids are wonderful models to study human brain development. And the advantages they present is whenever you have it from patients, they carry all the patient mutations. And the field is already thinking that, you know, there are differences between the mouse brain development and the human brain development. I mean, of course, there have to be differences because, you know, we're talking of a small brain as opposed to the huge expansion which the human brain has seen. So there are many differences in right from progenitor subtypes to, you know, the kind of diversity which we are seeing in the human brain. So in that case, organoids are a very valuable model because it's a human-specific model. It is coming from patients, so you can study patient-related mutations. So yes, so I think the field is slowly moving towards using human brain organoids to study these things.
Okay. And the next question is from Aarti Sharma. What changes occur in this neural layer in progeria?
I'm not, I don't know.
Yeah.
I think progeria is that, I think, where you age faster, right? I don't know whether there is any brain change in people with the condition of progeria. I don't know.
And the next question is from Vishek Bardhan. As shown by some study that FOXJ1 can be deregulated in case of central nervous system tumors like ependymoma, and FOXJ1 being an upstream regulator of LHX2. Does LHX2 also have any role in CNS cancer progression?
I'm not aware of any such study where CNS cancer progression LHX2 has been implicated. But perhaps people have not looked at, so I don't know. Maybe worth looking at such studies and looking whether there is any change in expression of LHX2.
Okay. And Shrikant Burgula wants to know, is LHX2 is required for BMP signaling?
Oh yes, required for BMP signaling. I don't know that, but there is so there is a possibility that LHX2 may have some crosstalk with the BMP signaling, BMP and the Wnt signaling, because the hippocampus actually, where it is made, the very adjacent structure to the hippocampus is called hem, which patterns the hippocampus, and the hem is BMP and Wnt rich. And this is study from Professor Shubha Tole's lab in 2008 when she published a very landmark science paper where she showed that, you know, this hem is very important regulator of the formation of hippocampus, and the hippocampus is formed in an LHX2 positive tissue. Wherever there is a hem which is Wnt and BMP rich and there is an LHX2 positive tissue, you will form the hippocampus in that. And she showed that if you have multiple hems, that is multiple centers with BMP and Wnt, and every time there is an LHX2 positive tissue, it will become a hippocampus. So she was able to make multiple hippocampus like that in the so I think there has to be some crosstalk, but I think we've still not worked it out.
Okay. And the next question is from Gaurav Bhardwaj. How a layer which is synthesized later pass through the preceding layer?
Sorry, could you repeat that again?
Yeah. How a layer which is synthesized later pass through the preceding layer?
Oh, that's a very nice question. So it's a very beautiful mechanism. What happens is these progenitors I showed you as balls. Actually, they're not balls. They are radial glia. So they have they have these glia-like processes which reach out to the PL surface, and the neuron which is just born actually uses this as a scaffold and migrates through this progenitor, which is a radial glia, migrates past the layer 6 and then goes and sits in the layer 5. So the progenitor itself acts as a scaffold for the just born neuron, and it crawls along this. It's a very nice migratory process. I think if you perhaps if you could see some videos, you'll be able to see that how it uses the scaffold and migrates past one particular layer of neuron and goes and sits on top of it.
Okay, so we have a couple of more questions. If we are okay, can we take more?
Yes,
if it's okay with you. Okay.
Yeah, no, it's completely fine.
So the Anushka Banerjee wants to know, do you see research using brain organoid models becoming more common, or do the cons outweigh the pros when compared to animal models? It seems that, especially with this kind of studies, it may be very advantageous.
So there are, as of now, where the technology is, we are not able to create entire brain structures. We are only able to create certain regions, say the forebrain or things, right? There is definitely an advantage. So when we talk of the brain, we are not talking of a single region, right? Like, as I was telling you in my talk itself, that the brain connects from one part to the other, like there is a crosstalk. And some of these connectivities are absolutely important for us to understand the function of the brain. So if your questions are very localized, specific to a cell type, yes, you can use brain organoids. But the moment you want to ask questions like how one region of the brain wires to the other, or what is a crosstalk, what is the connectivity, then you need a whole organism model system. So I think as of now, where we are, we need both the model systems to understand. Of course, the mouse is very, very useful to understand conserved mechanisms. That's why conserved mechanisms and to look at whole brain structures. But having said that, we need newer models because we need human-specific mechanisms. So we need brain organoids. So we need a bit of both. And let's see how the field progresses. And if we are able to generate whole brains, of course, ethics in question, so many things are need to be. It has to be kept in mind before we can go there. So right now, I think my own lab uses both model systems to study neurodevelopment.
And Mitu Mohan have asked, can you explain oncogenicity due to LHX2, and can this be used for gene therapy?
Oncogenicity, I'm not aware if LHX2 has any oncogenic potential. I'm not sure. I know it has multiple roles in different systems like the limb, retina, hair follicle, and everywhere. I'm not sure. I haven't come across a oncogenic role yet, or maybe I've not seen.
And Kaustav Mukherjee wants to know why LHX2 was removed only at embryonic day 11. Why not after that?
So we have. It all depends on the kind of Cree we use. So we used a Cree which was specific at 11.5. So now we are removing LHX2 at different time points in development, say 13 or 14, using different kinds of Crees. And we are looking at its roles. Because this transcription factor is expressed for development, we think that it has very specific roles at every embryonic age.
Yeah, and another question from Kaustav is PROX1 expression position changing in absence of LHX2?
So difficult to say because the whole hippocampus itself is, diminished. It's because LHX2 has a progenitor proliferation role as well, beyond fate specification, which means that the moment you remove LHX2 from progenitors, they exit the cell cycle. So that's why the cortex is shrunken, because the progenitor pool has diminished. So it is now difficult to say whether, it is because the cortex is shrunken and that's why your PROX1 expression levels have changed, or has changed position, because I think it's difficult to separate the two rules. So I would just leave it saying that, the cortex is shrunken, but the PROX1 levels are still there. In that, it's still specifying that, that region in the hippocampus.
Okay. So with that, we are done with almost all the questions.
Okay.
And I would like to ask one question.
Yes.
like with we know in the past decade we have very big projects have been undertaken, like the Brain Initiative and the Allen Institute's Brain Atlas, and then we have your then the development in the brain organized research and all. So with all these developments, huge amount of data is being generated with unprecedented insights.
Right.
So what according to you is the timeline, or like how far are we from having a complete understanding of this complex circuitry of the brain and understanding the cognitive function thereof?
We So, I mean, I think with every model system, we are uncovering more and more about the brain. I mean, few years back, perhaps, Drosophila, zebrafish, C. elegans were our model systems. And then suddenly we had mouse, studying this newer structures like the neocortex and all, using the mouse as a model system. And now we see that, the entire connectome of the Drosophila has been mapped. Right. And next, I think where we are going at is perhaps, looking at the entire mouse connectome. Right. So I think we are progressing with each bit. And yes, of course, there's a lot of data out there, but I think the data is all making sense now. I think the next 50 years probably are going to be very, very crucial for neuroscience.
Okay, fair.
Yeah, definitely. With a newer model system like the human organoids, or even people looking at ferrets as a model system. So I think we're slowly moving towards understanding that what makes the human brain, coming from other model systems. So
and apart from this, these days we are also having insights into like how the pathological forms of, say, the tau filaments actually do look like right in the brain samples.
right.
So we have really came a long way, and
Yeah, we've come a long way.
yeah, and I hope the idea of many institutes have been established with the idea of creating the whole brain, the human brain.
Yes.
So let's hope that, I don't know when it will get fulfilled, but at least if we have a clear understanding of the functioning of human brain, then I think it will help really a lot in tackling neurodegenerative disorders.
Yes, yes. I think both the basic mechanisms and the understanding in disease should go hand in hand. Yeah, I mean, one cannot do without the other. Like without basic understanding, we don't know what goes wrong in disease. And simultaneously, if we look at disease, we also understand that what perhaps could have been the mechanism by which it actually works during development. So now I think should go both hand in hand with, and all the kind of model systems we have. I think it's just, I mean, I don't know who said this. I don't know, but somebody said that the next 50 years, probably coming centuries for the neuroscience. So I think we have that's saying something, and that's very, very hopeful for all of us.
Okay. So thank you very much for taking out your time and agreeing to speak on our platform. It was really a very wonderful session, and thank you also for answering all the questions. And we do wish very best of luck to you. You have started your lab. You have started your lab recently, and we hope to get newer and newer insights about how brain organoids can actually aid us in understanding the human brain. So thank you again on behalf of the whole PVP team. I would like to extend a very
No, thank you to you, Dishant, and for organizing this wonderful series. And I think it's a very nice initiative because you can now hear so many different people speak on different topics, and it's a it's a boon in disguise, because now you can sit and probably learn so much about what's
exactly, and participants can also ask the questions freely. So it's really both way Indian situation.
Correct. Yeah. Yes, yes. And I think it's nice to know what's happening around India, because sometimes we only attend conferences in person, and we usually attend conferences in a particular field. So now with this, the advent of, as I said, it's a boon in disguise brought on by COVID-19. But I think it's a very nice series which we have started, Progress and Prospects in Biology. And I hope you continue this, and I come back at some point to talk about what my lab is up to with organoids and neurodevelopment. So I hope that, and thank you so much for the invitation.
Thank you so much for your kind words and appreciation. Thank you again for your kind words and appreciation, and we look forward to hosting you again in the near future.
Yeah. Okay. Thank you so much.
Thank you so much. And thank you all the participants for joining. And we do apologize for any technical glitches that has happened today, like YouTube. There was some problem on the streaming part. So what we will do is this whole of this talk has been recorded. So we will upload it on YouTube. So you can go and watch on our YouTube channel. And please do like, share, and subscribe to our YouTube channel. And next week we have two talk. There is a special talk on Saturday, 17th of October, by Professor Chandrima Shah from National Institute of Immunology and also the President of International Science Academy. And on next Sunday, Prabhan Chakravarti from Institute of Functional Genomics, Montpellier, from France will be talking. So we all look forward seeing you next week. So till then, you all take care and stay safe in this time. Thank you all.